Propagation is the foundation of many businesses within the diverse industry of horticulture. Propagation of plants can be achieved by many successful methods, but one of the most widely used and simplest methods of propagation is by cuttings. Along with seeds, cuttings remain the backbone of modern plant production. Cuttings require special tools and environments, but greatly increase the versatility of propagation. By making use of old as well as new methods of taking cuttings, we can increase the chances of successful propagation. In this program, we'll not only demonstrate the skills and techniques of growing plants from cuttings, but we'll also look at the science of cuttings so that we can come to understand how these techniques work. First, we'll take a look at some basic terms and definitions necessary for understanding the growth and development of a cutting, and go over the basic requirements for rooting. After reviewing the tools and special environments needed for cuttings, we'll see the proper techniques for collecting and sticking the cuttings. Let's now begin by defining some basic terms and concepts which will help us understand the science of propagating plants from cuttings. So what exactly is plant propagation? Plant propagation is the reproduction or multiplication of plants using seeds or other various tissues from a parent plant. To put it another way, plant propagation simply means making more plants. All of the many techniques of plant propagation fall into one of two main categories: seed and vegetative reproduction. In vegetative reproduction, also referred to as cloning or asexual reproduction, the plant is reproduced using a portion of vegetative tissues, such as a stem cutting. The resulting plants are genetically identical to the single parent from which the vegetative tissues were taken. In addition to cuttings, there are many other methods of vegetative propagation, such as division and layering, but the principles of asexual reproduction remain the same. For instance, the flowers of this rock rose have been developed and selected for their deep pink color. To ensure that the unique flower color remains unchanged during reproduction, this variety must be propagated using vegetative techniques such as cuttings. In reproduction by seed, also called sexual reproduction, male and female genetic material, or DNA, is combined through the process of pollination and fertilization. This type of reproduction produces variable results, with the new plants showing a mixture of traits from both parents. This seemingly random expression of inherited traits is called genetic variation. Genetic variation gives us the opportunity to develop unique plants, which may be of greater value. Through cross-fertilization, we can combine the desirable traits from two parents of the same species. The seed that is formed and the resulting plant is called a hybrid. The industry of horticulture is full of hybrid plants as well as many unique, naturally occurring varieties. If a plant breeder develops a very desirable plant, then it will often be reproduced using vegetative methods in order to retain all of the desirable traits. When a new variety has great value, such as a new color of rose, the new variety may be worthy of a plant patent. This would make any further vegetative propagation illegal unless you hold the patent rights. There are many criteria plant breeders look for when selecting these new plant varieties. Some of the most common criteria include resistance to disease and pests, cold hardiness, and drought tolerance. We use vegetative propagation for ornamental plants to retain characteristics such as flower size, fragrance, rare colors, unique form, or a long vase life for cut flowers. Fruit varieties are additionally selected for their flavor, storage qualities, and their ability to tolerate harvesting and shipping practices. Once we have selected a plant with the traits we want, then we will usually need to reproduce them using vegetative techniques. Again, this ensures that those qualities seen in the parent plant are also expressed by the new plants. Aside from retaining any desired characteristics, many plants are reproduced by cuttings because the seeds are not viable or simply not available. Even if the seeds are viable, some seeds have a lengthy dormant period which can be unpredictable and irregular. Still, other seeds have special requirements for germination that may be complicated or time-consuming. Cuttings have become a very common method of vegetative reproduction. Many cuttings can be taken over and over again from the same parent plant, known as the stock plant or mother stock. Stock plants, which are long-lived, can yield thousands of cuttings over their lifetime, giving us great return on our initial investment. Now let's take a look at the anatomy of a cutting. Most of the cuttings we will take will usually be stem cuttings. The basic stem cutting consists of a length of stem, leaves, a terminal bud, also called the apical bud, lateral buds, also called side buds, and nodes. Nodes are the joints of the stem where the leaves and side buds are attached. Leaves and buds are usually removed from the bottom nodes of the cutting. The segments of bare stem in between the nodes are called internodes. We'll refer to these terms again as we begin working with stem cuttings. Stem cuttings have long been classified into four main groups: hardwood, semi-hardwood, softwood, and herbaceous cuttings. With a few exceptions, hardwood cuttings are taken from deciduous shrubs or woody trees such as the fig. Hardwood cuttings are collected during the cold, dormant seasons from late fall to early spring. The previous year's growth is usually ideal for this type of cutting. Semi-hardwood cuttings are also taken from woody shrubs and trees, but the cuttings are collected during the warm summer months. The cuttings are taken from new shoots where the wood has partially matured. Softwood cuttings are often taken during the late spring season from the soft new growth of many woody plants. Softwood cuttings in general will root quicker than other types of stem cuttings. Herbaceous cuttings are taken from the soft, succulent growth found on flowering perennials and even some annual flowers. Herbaceous cuttings can be taken anytime as long as the plant is actively growing. These are probably the easiest cuttings to root and make a great confidence builder for beginning propagators. Stem cuttings are the most common type of cuttings, but leaf cuttings and root cuttings can also be quite productive. Leaf cuttings are made in a variety of ways depending on the plant type. Leaf cuttings can be made with just the blade of the leaf, as with Sansevieria, or the leaf petiole may need to be taken along with the leaf blade, as with Peperomia. Foliar embryos, although they are technically not true embryos, make for easy leaf cuttings since the new plants will form on the top side of leaves while still attached to the mother stalk. This gives the piggyback plant its curious leaf form. In contrast with leaf cuttings, root cuttings are less varied in terms of methods or techniques. Root cuttings are best if collected in the early spring and are effective with a variety of plants including this fig tree, Albizia julibrisson, Campsis radicans, and poplars. Now that we're familiar with the terms and concepts of cuttings, let's take a closer look at the rooting process for cuttings. The main goal with cuttings is to get the stem or other plant part to the point where it is self-sustaining. This requires the development of roots called adventitious roots. This process is also referred to as rooting. Adventitious roots are simply roots that arise from any part of the plant other than the seed embryo. So all roots that develop from cuttings would be considered adventitious roots. So what do our cuttings need in order to grow these new adventitious roots? Hormones are naturally occurring chemicals produced by all plants. A group of hormones called auxins are critical to the growth of adventitious roots. Auxins are now available in synthetic form for plants that are difficult to root. The naturally occurring auxins will accumulate in the base of a stem cutting due to the darkness and moisture of the soil. In addition to hormones, a suitable environment must be maintained during the rooting process. Until the cuttings develop roots, we need to provide most of the basic elements any plant would need to survive. This would include light, water, proper temperature, and aeration. Light is required to sustain the cutting during the rooting process. In greenhouse environments, sunlight stimulates the process of food production within the leaves. This fundamental process is called photosynthesis. At the same time, the sun raises the ambient temperature in the greenhouse. A misting system is needed to counteract the rising heat and possible dehydration of the cutting. Carbon dioxide is also critical to the photosynthesis process. Fortunately, carbon dioxide occurs naturally in great abundance and can be provided simply by good air circulation. Water is needed by the cutting to keep the plant rigid and to carry on basic life-sustaining functions. The evaporation of water from the leaf surface, known as transpiration, creates an important cooling effect on the plant. The water moving from the soil up the unrooted stem of the cutting provides only a small portion of the water needed by the cutting. Again, a misting system will usually compensate for the short supply of water. The proper temperature needed by cuttings will vary from plant to plant. The best temperature for rooting is usually lower than the ideal growing temperature for the rooted plant. Most plants will develop roots if provided with a temperature range between 18 to 25 degrees Celsius or 65 to 78 degrees Fahrenheit. Bottom heat increases our success with cuttings by stimulating cell division and promoting rooting. But the above ground portions of the cutting benefit by staying relatively cool. In general, optimum rooting temperature is about 10 degrees higher for the rooting zone than it is for the leaf zone. By lowering the normal growing temperature at the leaf surface, we can reduce the stress on the unrooted cutting. The ambient temperature can be reduced by venting, shading, and the evaporative cooling effect of misting. Oxygen is a well-known byproduct of photosynthesis. Most of us have heard that plants make oxygen, which animals need to survive. But plants also need oxygen for many basic functions, including the development of adventitious roots. Oxygen is very important during the process called callus formation. A callus is the scar tissue which forms at the base of the cutting, effectively healing the wound caused by our pruning shears. The buildup of callus is the first visible sign that the cutting is developing normally. In most cases, the new roots will grow out of this callus tissue. To enhance this callus formation, we need a rooting medium that is porous and allows oxygen to flow around the base of the cutting. Now that we're familiar with the basic rooting process, we're finally ready to stick the cuttings. A quick list of equipment we'll need before sticking the cuttings would include pruning shears, a plastic bag, or a large container for collecting the cuttings, rooting media and trays, or flats to support the cuttings, a misting system, and ideally some kind of bottom heat. Let's start out by taking cuttings of an herbaceous plant. Plants from the mint family are usually easy to root, and stock plants are also easy to find. They can be recognized by square stems, strong scented leaves, and flowers that are arranged in whorls encircling the stem. This spearmint is easily propagated by cuttings and should root quickly in 2 to 3 weeks. With herbaceous cuttings, we want to select stems that are thick and sturdy. Once we've collected enough stems, we'll spray a little water over them to keep them from wilting. If possible, keep the stems out of direct sunlight. The rooting medium we're using is 2 parts perlite mixed with 1 part peat moss. We'll use a plug tray with deep cells and fill it to the top with our rooting medium. Wet the medium after it has been added to the plug trays. Now we're ready to stick the cuttings. Each length of stem should provide us with many cuttings. As a general rule, each cutting should have 2 sets of nodes stuck into the medium and 2 sets of nodes above the medium, with the leaves still attached. So when we select the piece of stem for our cutting, it should have at least 4 nodes if possible. Cut off the stem just below the last set of nodes. The bottom nodes are typically prepared by stripping all the leaves off by hand. This speedy technique creates a large wound at the base of the stem where callus tissue will form and eventually grow roots. Leaves can also be removed with a pair of shears, but be sure to cut the leaves off close to the stem. The upper leaves can also be reduced by cutting them in half. This is not needed for mint, but it's an important technique for cuttings with large leaves. Also remove any flowers or flower buds which might drain the energy out of the cutting. Now stick the cuttings into the medium, just deep enough to give the plant support and far enough to cover the two lower nodes. Do not press the cutting to the bottom of the tray. This can cause the stem to dry out. As the cuttings are pushed in place, try to keep the leaves arranged so that they don't cover the stem tips of nearby cuttings. This is where the new shoots will sprout out. We can also prevent diseases by positioning or even pruning the leaves so that they don't come in contact with the medium. Once the tray is filled, it is ready for the misting system. Quick rooting herbaceous cuttings only need to be misted 3 or 4 times per hour, depending on the temperature. The plants should only be misted during daylight hours and never during the night. Another type of stem cutting we've mentioned is the hardwood cutting. Hardwood cuttings should be collected during the cold, dormant months. The leaves of this edible fig, Ficus carica, have fallen off, indicating it is now dormant. When harvesting the bare branches, select stems that are about 1/2 to 3/4 of an inch in diameter. Make the cuts just below a node, with each cutting measuring about 9 inches in length. Bundle the hardwood cuttings together and let the bottom ends of the cuttings stand overnight in a solution of rooting hormone. After the hormone treatment, it's helpful to let the pruning wounds callus over before planting. Store the cuttings in a moist container over wood chips or peat moss for about a month or until well-defined callusing is noticeable. After callusing, be sure that the cuttings are planted the right side up, with the bud tips pointing upward and the leaf scar positioned just below the bud. If rooting is successful, these buds will begin to push out leaves in the spring. The fig tree is also a good plant for propagating by a root cutting. Most root cuttings should be made in early spring when the roots still have plenty of food stored from the previous dormant months. Cut out sections of medium-sized roots 1/4 inch to 1/2 of an inch in diameter. Since there are no buds to indicate which direction is up, we should cut the base of each root cutting with a diagonal cut, and then remember to point all the diagonal cuts in the downward position when sticking the cuttings. The top of the cutting may need to be recut flat so that all the tops are level and all the bottoms are pointed. We'll make each of these root cuttings about 4 inches long. As with hardwood stem cuttings, the fig's root cuttings should be stored in a moist box for about a month. The root cutting can then be planted in rooting media or directly into the field, where the entire root piece is covered with soil. Aside from stem cuttings and root cuttings, leaf cuttings are also an easy propagation method, especially for some tropical plants and succulent plants. With many succulents, such as these donkey tail, The leaves can be broken off and simply set directly on the surface of the potting soil. Another type of leaf cutting, called the leaf blade cutting, only uses segments of the leaves for rooting. One of the most common examples of the leaf blade cutting is made using Sansevieria, commonly called the snake plant or mother-in-law's tongue. 3 to 4 inch sections are sliced up, giving the downward or distal end of each leaf a slanted cut. This technique, as shown earlier with root cuttings, will help us maintain proper orientation when sticking the cuttings. Using a standard propagation medium, push the cuttings halfway into the rooting medium, remembering to point the slanted end of the leaf downward into the medium. These leaf blade cuttings will usually root in 8 to 10 weeks. A variation on the leaf blade cutting is the midrib cutting, used with these Streptocarpus. After removing an entire leaf, use a razor or sharp knife to cut out the middle of the center leaf vein known as the midrib. Place the halves in a rooting medium buried about 1/2 inch. These leaf cuttings will need to be set under mist for about 2 months. These pups are now ready to be divided and transplanted. Another type of leaf cutting is the leaf petiole cutting, in which the entire leaf, including the petiole, is used. This peperomia is one of the many tropical plants that will root easily with this method. The petiole is pushed into the rooting media until the bottom of the leaf blade contacts the media. New roots and pups will emerge in a month or 2. The rooted leaves and pups can then be transplanted into pots. African violets are also propagated using this technique. An unusual type of leaf cutting is the foliar embryo. As mentioned earlier, foliar embryos are technically not true embryos, but they do make easy leaf cuttings. The foliar embryo of Tolmia menziesii, commonly called the piggyback plant, sprouts a new plantlet or pup from the base of the leaf blade. These small plants are often already formed on the adult leaf, complete with tiny roots. They merely need to be set on some potting medium in order to sprout roots. We'll use the direct stick method, which skips the need for transplanting the rooted cuttings. With direct stick, we use the pot that the plant will eventually be sold in. In the case of the piggyback plant, we're making a hanging basket for use as a houseplant. We'll use regular potting soil instead of the rooting media, since the cuttings will only need to be misted for a short time. Now that we're familiar with the basic methods for sticking cuttings, let's see how to grow on and transplant the cuttings once they have rooted. While the cuttings are developing roots, we should be checking on them every week or so to see if there is any callus formation at the base of the stem. Slow callus formation is often a sign that the media is not draining well, or the duration of the mist may be too long. The duration of mist should be long enough to cover the leaves evenly, usually between 3 to 5 seconds. The interval between misting should be long enough so that most of the water has already evaporated from the leaf surface. This misting interval will also depend on the type of plant being propagated. In warm greenhouses, misting intervals can run anywhere from one misting every 10 minutes to one misting every 30 minutes. If our misting program is scheduled correctly, we should soon observe roots growing out of the bottom of the tray. We can also check for rooting by giving the cutting a gentle tug from the leafy top of the cutting. If the cutting slips easily up and down, then the roots most likely haven't developed yet. While the roots are forming, the cuttings need to be regularly inspected for disease problems. Remove any signs of diseased tissue such as dark spots, black leaf tips, or grayish-white threads. Keeping the rooting tray clean will also help prevent disease infection. Pick up any rotting or fallen leaves and throw out cuttings that are obviously not going to take. As we mentioned, starting out with a well-draining, sterile soil will prevent many disease problems. Programming the mist schedule so that plants are not overwatered will prevent disease as well as algae growth. In addition, providing good air circulation and bright light will help control many diseases. If fine-tuning our horticultural practices is not effective, then diseases may be prevented by spraying or drenching with fungicides. Once the cuttings have rooted evenly throughout the tray, then they should be taken off the misting benches and watered only when dry or nearly wilting. Watering 1 to 3 times a day is usually enough. Transplant the cuttings into a potting mix that has better water retention than the rooting media. Equal parts of peat moss and perlite will make a good potting medium for most plants. Water the cuttings into their new pots, even if the medium was already moist. This ensures good contact between soil and roots and also reduces transplant shock. If we are transplanting the rooted cuttings directly into the field, make sure they have a hardening off period. Hardening off gives plants that have lived inside the greenhouse a transition period to get used to the harsher conditions found outside of the greenhouse. The plants are simply set outside for a week or two before transplanting into the field or landscape. Hardening off reduces the stress of that final move into the field environment, where the plant will complete its growth and development. In this program, we've looked at many aspects of propagation of plants by cuttings. We've studied the basic anatomy of cuttings as well as the process of callusing and root development. We also went over the basic horticultural practices that will ensure success and finished by showing how and when the cuttings should be transplanted. We hope that the combination of botany, biology, and horticulture covered in this program has sparked the interest of our viewers to continue studying and experimenting with the many techniques of plant propagation.